My name is Ch13 Anderson, and I am presenting on Section 7 of Chapter 13, Long-Term Potentiation. Think of the neurons like people walking through a forest. The first time you walk down a path, it's hard, but the more you use it, the clearer it becomes. That's what happens in the brain with LTP. The repeated activity strengthens the connections, making it easier for the signals to travel the next time. LTP is basically the cellular mechanism behind learning and memory, including conditional learning. Consider Pavlov's experiments with the dogs. Conditional learning is when we learn to connect the two things like a bell and food. LTP is the brain's mechanism that makes the learning stick. It strengthens the neurons that fire together, so the association just becomes stronger and long-lasting. Long-term potentiation, or LTP, is when a neuron becomes more excitable or more likely to respond to a certain input after that input is repeatedly active at a high rate. Basically, it's how neurons get better at talking to each other after practice. In the brain, the hippocampus, which helps with learning and memory, gets most of its input from a region called the entorhinal cortex. One important pathway, called the perforant pathway, becomes more active either when we experience things in our environment or when it gets a strong burst of electrical signals. This activity helps strengthen the connection, which is the basic idea behind LTP. This figure shows how long-term potentiation is studied in the hippocampus. In part A, we see a diagram of how different parts of the hippocampus connect. Scientists send small bursts of electrical activity through these pathways to imitate what happens when we learn something new. And in part B, we see the results. These are recordings from a part of the hippocampus called the dentate gyrus. After the stimulation, the signals become stronger, showing that the neuron connections were strengthened. That's long-term potentiation in action. A good way to think about it is like muscle training at the gym. The more you work out the muscle, the stronger it gets. This figure is showing how the speed of stimulation affects whether long-term potentiation happens. When the axons are stimulated rapidly, the small signals they send, called EPSPs, start to add up or summate. This builds up enough electrical activity to strongly activate the postsynaptic neuron, kind of like quickly stacking blocks until they reach the top. When this happens, the neuron becomes depolarized enough for LTP to occur, and that just means the connection gets stronger. If the axons are stimulated too slowly, those signals are going to fade away before the next one arrives, so they won't get a chance to add up, and the neuron won't reach the activation level that it needs to. As a result, LTP doesn't happen. You can think of it like pushing somebody on a swing. If you push it fast enough, the push is going to build up on each other and the swing goes higher and higher. That's LTP. NMDA receptors are really important for strengthening the connections between neurons in the process of long-term potentiation. It happens when the neurotransmitters bind to receptors on a dendritic spine that's already depolarized. In other words, the synapse has to be active and the postsynaptic neuron has to be ready to receive the signal at the same time. The NMDA receptor acts kind of like a special gate for calcium ions. It only opens when two things happen at the same time. Glutamate has to be present and the postsynaptic membrane has to be depolarized. When those conditions are met at the same time, the calcium flows in, and that calcium entry is what helps strengthen the connection between the two neurons. This figure shows the conditions for long-term potentiation. The synaptic strengthening only happens when both the presynaptic and the postsynaptic cells are active at the same time. So continuing with the NMDA receptors, when calcium ions enter through the NMDA controlled channels, that's the key step to actually triggering LTP. The calcium entry sets off a chain of changes inside the neuron that make the connection stronger and more efficient over time. There's also something called a dendritic spike. That's when the depolarization in the cell body spreads back into the dendrites. This backwash helps reinforce the activity in the synapses that were just used. Now here's when it gets interesting. If a weak synapse and a strong synapse are active at the same time on the same neuron, the weak one actually gets stronger. This is called associative long-term potentiation. This slide shows how the NMDA receptor acts like a gate that needs two keys to open, one chemical and one electrical. When the postsynaptic membrane is at rest, a magnesium ion sits in the NMDA channel and blocks it. That means the calcium ions can't get through at all, even if glutamate is present. But when the membrane becomes depolarized, that magnesium ion is kicked out of the way. Now, if glutamate binds to the receptor, the channel can finally open and the calcium ions can flow into the dendritic spine. So both conditions have to happen at once. The glutamate has to attach and the neuron has to be depolarized. Only then does the calcium enter, and that calcium entry is what starts the process of LTP. This image is showing what's happening during associative long-term potentiation. When the strong synapses are active enough to trigger an actual potential, so the neuron actually fires, that signal travels back through the dendrites as what's called a dendritic spike. This spike depolarizes the membrane in nearby dendritic spines, basically priming those NMDA receptors. Now, if a weaker synapse is active at the same time, its NMDA receptors are ready to open, letting the calcium in and strengthening that weak connection. So in the photo, you can see how the activity from the strong synapse spreads out and helps lift up the weaker ones. AMPA receptors play an important role in strengthening the synapses during LTP also. When the calcium ions enter the dendritic spine, they trigger the movement of more AMPA receptors into the postsynaptic membrane. This makes the neuron more sensitive to the glutamate, strengthening the synaptic connection. The enzyme CAMK2 helps control this process. It's activated by the calcium and is very important for maintaining these changes that's occurring. This slide shows the steps of synaptic strengthening during the long-term potentiation process. Like we talked about previously, first, when the conditions for LTP are met, calcium ions enter the dendritic spine through NMDA receptors. These calcium ions activate the enzymes inside the spine. Next, the activated enzymes cause AMPA receptors to move into the postsynaptic membrane. And finally, having more AMPA receptors strengthens the synapse, making it more responsive. This slide explains how long-term potentiation changes both postsynaptic and presynaptic parts of a synapse. LTP can change the size and the shape of the dendritic spines and even cause new ones to grow. LTP can also affect the presynaptic side. Nitric oxide play a very important role at this point. When it's produced in the dendritic spines, in the hippocampus, it diffuses to the terminal buttons and helps trigger changes that support the LTP process. This slide focuses on the role of protein synthesis and long-term potentiation. LTP happens in stages. The first stage, called early LTP, happens quickly and doesn't last long. But for the changes to last, we need the neurons to make new proteins, and that's what we call long-lasting LTP. One important protein involved is called PKM-zeta, which helps maintain and stabilize the long-term changes in the synapse. LTP doesn't only happen in the hippocampus. It also occurs in other areas of the brain. There's also an opposite process called long-term depreciation, or LTD, where the connections are actually weakened instead of strengthened. This figure shows how dendritic spines grow after LTP. These two photon microscope images capture a segment of a dendrite from a CA1 pyramidal neuron taken before and after electrical stimulation that produced LTP. You can see that after the stimulation, the dendritic spines became larger and more numerous. This shows that the long-term potentiation doesn't just strengthen existing connections, it can actually lead to the growth of new ones. This figure is highlighting the role of PKM-zeta in maintaining long-lasting, long-term potentiation. PKM-zeta is a protein that helps keep the synapses strong after LTP has already been established. It works by maintaining the increased number of AMPA receptors in the postsynaptic membrane, which keeps the synapse responsive and stable over time. Without the PKM zeta, these strengthened connections can fade, meaning the memory or the learned association would weaken and weaken. This slide is introducing the long-term depression that I was talking about earlier. It's kind of the opposite of long-term potentiation. LTD plays an important role in learning. It helps the brain fine-tune its connections by weakening synapses that aren't being used as much anymore. In associative LTD, synaptic inputs are active when a postsynaptic membrane is only weakly depolarized or even hyperpolarized. This causes a decrease in the number of AMPA receptors in the dendritic spines, making those connections weaker and weaker. This graph shows a relationship between the stimulation frequency and the strength of the synaptic responses, measured as the slope of the EPSP. On the x-axis, we have the frequency of stimulation of Schofield collateral axons, and on the y-axis, the percentage change in the EPSP slope. When stimulation is frequent and strong, the EPSP slope increases. This is long-term potentiation, which strengthens synapses. When stimulation is weak or infrequent, the EPSP slope decreases. This is long-term depression, or LTD, which weakens synapses. So to wrap things up, long-term potentiation and long-term depression are key ways that the brain uses to change its connections in response to the things that we experience. LTP is like frequently traveling down a trail and it becoming wider and clearer. LTD is the opposite. It's like a path falling into disuse, getting overgrown and less clear. The protein PKM zeta is like the maintenance crew, keeping the most important trails open. Together, the processes allow the brain to learn, adapt, and store memories by shaping which neural paths are strengthened and which ones fade away. Thank y'all for listening. I hope y'all learned something.